Hello friends, welcome back to the channel, and today's video is again about Krebs cycle. So what is Krebs cycle that we have seen in the previous video? In this video we are going to just concentrate on the various enzymes which are involved in Krebs cycle, and in addition to that we will see its energetics, that how it is helping us to generate energy. So we have already seen glucose get converted to pyruvate, pyruvate to acetyl coenzyme A, and this acetyl coenzyme A undergoes this cyclic pathway to produce citrate, isocitrate, alpha Kg, then succinyl CoA, then succinate, then there is fumarate, then there is malate, oxaloacetate, which is again reused. So now let's see the enzymes one by one here. So what happens in step 1? This acetyl coenzyme A fuses with oxaloacetate to produce citrate. So there is formation or there is synthesis of citrate. So the enzyme which is related to this step is very easy, that is citrate synthase. Means the enzyme which is responsible for synthesis of citrate. Now this enzyme is highly regulated. It is a very important enzyme for this Krebs cycle pathway, and this enzyme is having allosteric inhibition. It is having certain pockets through which it gets feedback that whether it should work or it should stop working, and how it happens. So let's see that Krebs cycle, when our body needs Krebs cycle, when there is lack of energy, and how the citrate is going to get the signal for that. This enzyme is going to get the signal for that. So when there will be higher amount of ATP in the body, there are enough amount of NADH, there is enough quantity of citrate, there is enough of succinyl CoA. So what will happen? It means that body is having sufficient energy. There is no requirement of further breakdown of acetyl coenzyme A. So for that purpose, these molecules are going to act as allosteric inhibitors of this enzyme, and they will tell this citrate synthase that yes, stop working now. There is enough of ATP, there is enough of NADH, there is enough of citrate, so don't make more citrate. And in this way, this function of this enzyme will be inhibited and the Krebs cycle will stop. And when there is now, it is not that we just want to inhibit it, we need to activate it also, we need to stimulate it also. So who is going to stimulate this enzyme? So as we know that ATP, it gets converted into ADP plus phosphate, inorganic phosphate. Now when there is less of ATP, there will be more of ADP. Means when the ATP will break down, there will be formation of ADPs. So this ADP, when it will be more in concentration, it means that citrate synthase will get the signal that ATPs are lesser. You need to produce more citrate so that there will be generation of more energy. So this ADP is going to act as a stimulator for this enzyme citrate synthase. So this is how this enzyme is regulated, which is the first enzyme for this Krebs cycle pathway. Now let's see the second step. What happens in second step? Citrate undergoes isomerization to produce isocitrate, and here the enzyme involved is Aconitase. ACOTINITASE, Aconitase. This enzyme is very important for some poisonous for some poisonous chemicals. For example, rat poison acts on this enzyme, that is fluoroacetate. It acts on this enzyme, it blocks this enzyme, it inhibits it, and this Krebs cycle is inhibited, and it leads to cell death, and eventually there is a death of the living organism. So this is an important enzyme, Aconitase. It is such a small step in Krebs cycle, and it is having such an important function. Then the next step is conversion of isocitrate to alpha ketoglutarate through decarboxylation. There is removal of one CO2, and this step is regulated by an enzyme, or it is catalyzed by the enzyme isocitrate dehydrogenase. Now remember one thing, when there is dehydrogenase, there is involvement of NAD plus, which gets converted into NADH. So In this step, one NADH is generated. Then the next step is conversion of alpha-ketoglutarate to succinyl coenzyme A. Now this step is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase. So it is quite similar to the name of the substrate, so I think it will be easy to recall, and we have already seen how to recollect the name of substrates. So this alpha-ketoglutarate dehydrogenase, again there is dehydrogenase, it means there will be more of NADH, some energy is going to be produced in this step again, and NAD plus will be utilized. Now we have got succinyl-CoA. This succinyl-CoA gets converted into succinate in the presence of enzyme succinyl-CoA synthetase. One important thing that I want to discuss here about this enzyme alpha-ketoglutarate dehydrogenase is that it acts as a cofactor in histone demethylase, and we know that the DNA, the coil structure of DNA, it is wrapped around the histone proteins, and if there is any alteration in this enzyme, what happens? This overall process of gene expression and cell growth, that all gets hampered, and due to that there is mutation, or the gene expression gets hampered, and that leads to uncontrolled cell growth, and that may lead to further gliomas. The cancerous or tumorous growth is observed. So this is an important enzyme in the formation of cancerous cells in the body. So this is a second, I think, like a small step of Krebs cycle, and it is having such an important role in the mechanism of life-threatening disease, that is cancer. Now let's see the next step, that is succinate we have got here now. This succinate gets converted into fumarate, and for that purpose we need an enzyme, that is succinyl dehydrogenase. Now in this step, FAD is utilized to produce FADH2. And one thing that I forgot here to include is that during this previous step, this conversion of succinyl-CoA to succinate, in this step a molecule of GDP is utilized to produce GTP, and this GTP is, now this fumarate, in the presence of water, it forms malate, and the substrate is fumarate. So the name of the enzyme is fumarase, and this fumarase converts fumarate to malate, and further the last step, that is formation of oxaloacetate from malate. So this step is catalyzed. So in this step again this malate dehydrogenase is there, and when there is dehydrogenase, obviously there will be involvement of an NAD plus to produce NADH. So in the last step, now we have got NADH, and the substrate produced here is oxaloacetate. This oxaloacetate will be reutilized again in the Krebs cycle to combine with the next molecule of acetyl coenzyme A, and the cycle will continue. So now let's just summarize the energetics of this process. So we can see that from glucose we get 2 pyruvates, it means we get 2 acetyl-CoA. The cycle will be repeated twice, it means we are going to get 2 CO2, 2 CO2 here, 2 NAD plus NADH will be produced, 2 NADH will be produced, 2 GTP will be produced, which is going to give us 2 ATPs. 2 FADH2 will be produced, then there will be 2 NADH again here. So through cyclic metabolism of one molecule of glucose in Krebs cycle, the outcomes which we are getting is we are going to get 4 CO2s, we are going to get 2 plus 2 plus 2, that is 6 NADH, 2 FADH2 and 2 ATP. So that will be the total outcome of this Krebs cycle pathway. I hope this explanation helps you to understand this important cyclic pathway. Thanks for watching the lecture, and Keep watching the channel. Thank you.